Hello, good evening, madam.
Hello, sir. Good evening.
Good evening. So today you are going to take about systemic bacteriology, I think, no?
Yes, sir.
Yeah, this is the last chapter in
Chapter, yes, sir. Then
first year.
whole this syllabus will be over for first year.
Oh, yeah.
Mhm.
So now we can continue with second year.
Yes, sir.
Yeah, yeah, madam. Please proceed, madam.
I'll proceed. All right.
Yeah, please proceed.
So, hello everyone. Welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's topic is about systemic bacteriology. So, what exactly is systemic bacteriology? It focuses on studying bacteria that can cause infections
Madam,
throughout the body rather than hello.
it's recording, no? Yeah, please proceed.
All right. So, systemic bacteriology focuses on studying bacteria that can cause infections throughout the body rather than being restricted to a single organ or site. It involves understanding the structure, morphology, biochemical properties, its classification, pathogenicity, how they cause diseases, its clinical significance, laboratory diagnosis, treatment, and prevention. So, this knowledge is essential for medical laboratory, that is MLT students, because identifying bacteria accurately is the first step in treating infectious effectively. So, introduction. The human body is inhabited by thousands of different bacterial species, some of which living transiently, others in a permanent parasitic relationship. Likewise, the environment that surrounds us includes the air we breathe, water we drink, and food we eat is inhabited by bacteria, many of which are relatively avirulent and some of which are capable of producing life threatening diseases. So, the systemic bacteriology is aimed to improving your understanding of the important bacterial pathogens. It causes disease, pathogenesis, laboratory diagnosis, and treatment options, preventions, and control. So, we'll start with gram positive cocci. This slide introduces the first group, that is gram-positive cocci, which are spherical in shape, bacteria that appears purple under a microscope when stained using Gram's method. The purple color is due to their thick peptidoglycan layer that retains the crystal violet stain. Its important genera in this group include Staphylococcus, Streptococcus, and Enterococcus. So these organisms are a part of normal flora on skin, mucous membranes, and throat, but can become pathogenic under certain conditions. So understanding them is crucial as they can cause a wide range of infections from mild skin conditions to life-threatening systemic diseases. Asorius are widely distributed in nature and cause variety of severity in infections. There are about 33 species of staphylococci identified. Species which are of medical importance include S. aureus, that is Staphylococcus aureus, S. epidermis, and S. saprophyticus. Its cultural characteristics. So, Staphylococcus aureus, a major pathogen among gram-positive cocci, its morphology, it appears as grape-like clusters under the microscope. It is a facultative anaerobe that can grow with or without oxygen and is catalyst positive. Normally, it resides on the skin, nasal cavity, and mucous membranes without causing harm. However, when it breaches the skin barrier or when immunity is low, it can cause infections. It is known for its ability to produce a wide range of enzymes and toxins, which make it one of the most versatile pathogens. So the diseases it produces, it has the ability to multiply and invade tissue, production of extracellular enzymes and toxin, as well as it will produce beta hemolytic colonies on blood agar. Hemolysis. So the ability of bacteria to cause hemolysis, which is nothing but breakdown of red blood cells when cultured on blood agar plate. Hemolysis is an important diagnostic features. There are three type of hemolysis: alpha hemolysis, beta hemolysis, and gamma hemolysis. Alpha hemolysis, it is known as partial hemolysis. It produces a greenish discoloration around colonies, for example, Streptococcus pneumoniae. Beta hemolysis, it is complete lysis of red blood cell, produces a clear colorless zones, for example, S. aureus and S. pyogenes. Gamma hemolysis, it means no hemolysis will occur, no change in the medium. This property helps differentiate species with Staphylococcus aureus and Streptococcus species. So antigenic composition, this slide highlights the cellular and surface structures responsible for its virulence. So capsule, teichoic acid, protein A. So teichoic acid, it helps in adhesion to host cell and can activate complement. Protein A, it binds to the Fc region of IgG, immunoglobulin G, preventing opsonization and phagocytosis. These antigens not only protect the bacteria but also targets for laboratory identification and potential vaccine research. Peptidoglycan provides structural integrity and can stimulate immune response. Capsule, it prevents phagocytosis by immature immune cells. Coagulase, it causes the clumping of non-capsulated strain when it is mixed with a solution containing fibrinogen, for example, plasma. Enzymes and toxins produced by S. aureus. So coagulase, it clots plasma to protect bacteria from host defense. Cytotoxins, there are three types of toxin: alpha, beta, and gamma toxins. Hemolysins, it causes lysis of RBCs, that is red blood cells, of many animal species, which demonstrated as beta hemolysis on blood agar media. Exfoliative toxin, that is ET and ETB, it causes scaled skin syndrome mainly in infants. Toxin shock syndrome, TSS1, it acts as a superantigen leading to massive cytokine release causing fever, rash, shock, and multi-organ failure. Enterotoxins, it causes food poisoning. They are heat stable and lead to rapid onset vomiting and diarrhea. These toxins are responsible for many non-purulent diseases of S. aureus. Other toxins and enzymes include leukocidine, proteinase, catalase, staphylokinase, lipase, and hyaluronidase. So staphylokinase, it dissolves clot and helps the bacteria spread. Lipase, it helps colonize sebaceous area of the skin, and hyaluronidase, it breaks down hyaluronic acid in connective tissues, which enhance its spread of infection. Disease produced by S. aureus. So there are three types of disease which is produced by S. aureus. First is superficial infection, second is deep-seated lesions, third is bacteremia. In superficial infection, it causes folliculitis, carbuncles, boils, style, mastitis, abscesses formation. In deep-seated lesions, it causes osteomyelitis, septic arthritis, endocarditis, meningitis, endocarditis, and bronchopneumonia, emphysema, etc. Third is bacteremia with multiple abscesses in tissue. It will outbreak of hospital wound infection commonly occurs due to antibiotic resistant staphylococci. So localized skin infection, it will also include boils, impetigo, etc. Wound infections, it can include post-surgical and traumatic infection, and in deep-seated lesions, it will also include cellulitis, abscesses. It shows the diversity of infections from minor to severe depending on host immunity and bacterial virulence. Toxin mediated staphylococcal diseases. Food poisoning, it is generally caused by preformed enterotoxins. It will occur because from indigestion of preformed enterotoxin in contaminated food which is improperly cooked or kept unrefrigerated for long period of time. Its source of contamination will include the hand or nose of an uncooked person, food handlers, carriers. Types of food involved in staphylococcal food poisonings are carbohydrate-rich foods, which includes cakes, pastries, milks, etc., bakery products. It's it will take 1 to 8 hours, which will cause followed by nausea, vomiting, diarrhea, and general malaise with no fever. These are the symptoms which will cause food poisoning. Second is toxic shock syndrome. It is due to TSS1. It is associated, first described in menstruating women using tampons. The syndrome also occurs with wound or localized infection, which abrupts onset of fever, vomiting, diarrhea, muscle pain, rash, heart, and renal failure. It may occur in severe cases. Third is staphylococcal scald skin syndrome, that is SSSS. It is happening due to exfoliative toxin. It highlights how SORS causes diseases not only by direct tissue invasion but also by producing potent toxins. The syndromes occurs generally in babies and young children and characterized by large areas of desquamation of the skin and bluey formation. Scale skin syndrome. This image showing scale syndrome in a 6-year-old boy. Laboratory diagnosis. So, how SORS infection are diagnosed in the laboratory? First is specimen collection. It depends on the infection site, which include pus, wound swabs, sputum, CSF, blood and urine examined, etc. Then they'll do microscopy, that is gram stain, which shows gram-positive cocci in grape-like structures. Culture, it grows readily on nutrient agar media, blood agar showing beta hemolysis and mannitol salt agar, ferments mantle producing yellow colonies. Further, they'll do biochemical test, catalase positive, coagulase positive, antibiotic sensitivity testing, which is essential to detect SORS. This diagnostic approach ensures correct identification and appropriate therapy. Here is a microscopic image of gram-positive cocci, coagulase negative staphylococci. So, in this image, it likely indicate continuation with gram-positive cocci, which focus on other beside SORIAT. It also serve as a reminder that gram-positive cocci include not only Staphylococcus species but also coagulase negative Staphylococci, Streptococcus species, and as well as Enterococcus species. Each group has its own clinical significance and diagnostic method. Coagulase negative Staphylococci. These species do not produce coagulase enzymes. Its example includes Staphylococcus epidermidis and Staphylococcus saprophyticus. They are part of normal skin flora but can cause hospital-acquired infections, especially in patients with indwelling catheters, prosthetic valves, or implants, shunts, or surgery. Urinary tract infections, UTIs, particularly caused by Staphylococcus saprophyticus in young women. They are usually less virulent than SORS but important in nosocomial settings. Asaurus saprophyticus, it is commonly associated from animals and their carcasses. It is resistant to antibiotic novobiocin. It is a characteristic used in lab to distinguish it from S. epidermidis. It is the second most cause of UTI infection after E. coli in young women, accounting 10 to 20% population. It also causes soft tissue infections. Streptococcus. The genus Streptococcus, another major group of gram-positive cocci. They appear as chains or pairs of spherical cells. They are catalyst negative, unlike staphylococci. Streptococci are classified based on hemolysis and lancified groupings, that is A, B, C, D, etc. They are responsible for a wide range of infections, including pharyngitis, scarlet fever, rheumatic fever, and neonatal infections. So, more than 30 species are still identified. Those most are facultative anaerobes and found in water, dust, milks, and dairy products. This is a diagrammatic representation of Streptococcus. This scanning electron microscope image of Streptococcus demonstrates a morphology which is round cells arranged in chains or pairs. This visual representation helps students appreciate the structural arrangement and contrast its Staphylococcus clusters. Classification of Streptococcus. So, it is a heterogeneous group of bacteria according to their oxygen requirements. Some are aerobic and some are anaerobic according to their action on red blood cell in blood agar plate. Beta hemolysis Streptococci, complete hemolysis will be seen, clear zone around the colonies on blood agar. Example include S. pyogenes and S. agalacti. Alpha hemolytic streptococci, it will show partial hemolysis. Example include viridian streptococci and S. pneumoniae. Third is non-hemolytic or gamma streptococci. No hemolysis will be seen, no change in RBC cell. Example is enterococci. Based on serological activity of cell wall polysaccharide, it is classified according to lancified group. Group A streptococci, it will include pharyngitis, cellulitis, rheumatic fever, glomerulonephritis, which is S. pyogenes. Group B streptococcus, it will include neonatal sepsis and meningitis. For example, S. agalacti. Group C, S. equisimilis. Group D, enterococci. Group H, S. sanguis. Group K, S. salivarius. Usually, we'll study deep about S. pyogenes, enterococci, and equisimilis and agalacti. Streptococcus pyogenes. So its habitat, it is known as group A streptococcus, that is GAS, the most important human pathogen which was causes diseases including suppurative conditions, that is skin infections, throat infections, systemic infection. Non-suppurative sequel, about 5 to 15% of normal individual will harbor this bacterium in their respiratory tract without sign of any diseases. Its habitat is human throat and skin. Infections virulence factors include M protein, streptolysin, streptokinase, and hyaluronidase. Disease, it will cause pharyngitis, impetigo, scarlet fever, all this, etc. Rheumatic fever. Complication is post-streptococcal glomerulonephritis. So, virulence factor of group A streptococci, M protein, antiphagocytic, major virulence determinant, hyaluronic acid and capsule, it will prevent opsonization and inhibits phagocytosis. Exotoxin such as pyrogenic toxin, it will cause the rash or scarlet fever and systemic toxic shock syndrome. Invasions, streptokinase, streptodornase, that is GNSB, hyaluronidase, and streptolysins. It will lyse red and white blood cells, and streptokinase, it will break down fibrin clots. Pathogenesis. So, pathogenesis diseases due to toxin is of 3 to 4 types. First is streptococcal throat, that is skin infections. Second is diseases due to invasion. Third is non-suppurative sequel. So we'll start with first diseases due to toxins. So streptococcal sore throat infections and follicular tonsillitis, that is pharyngitis, it is characterized by enlarged toxins with purulent exudates. It will cause high fever and enlarged cervical lymph nodes. Impetigo, a local infection of skin characterized by formation of blisters. Red color blister will occur on skin which will break, leaving surface covered with pus or crust. Entry and colonization. Bacteria colonize the throat or skin via adhesins, that is M protein and lipoteichoic acid. Invasions of tissue. So enzymes like hyaluronidase and streptokinase help them penetrate tissue barriers. It will diffuse rapidly, spreading infections that will involve lymphatics with minimal local suppression. Infection can extend to the bloodstream. Puerperal sepsis, infection of the uterus after delivering or abortion. It will lead to endometriosis and it is associated with bacteremia. Soft tissue sepsis, it will include wound infection, that is cellulitis, lymphadenitis, necrotizing fasciitis. It is complicated due to streptococcal septicemia. Third is acute bacterial endocarditis. The organism reaches the heart valve through bloodstream. It will cause complication because any of the primary lesions will complicate this infection, that is known as acute bacterial endocarditis. Third is non-suppurative sequel. It occurs in post-streptococcal disease after 1 or 4 weeks following a primary inadequately treated group A streptococcal infection of the skin and respiratory tract. Acute glomerulonephritis, that is AGN. This condition is due to antigen antibody complex deposition on the glomerular basement membrane, that is GBM. The majority of patients will recover completely with this acute glomerulonephritis. However, few may die or pass to chronic GN and renal failure.
Excuse me, madam.
Rheumatic fever. Hello? Yes, sir?
Madam, it's showing on top in your laptop or something recording is going on. Just check.
Recording is not showing, sir.
Not showing.
No, it is just showing that you are screen sharing.
No, madam, it's recording actually. Okay, madam, you continue.
All right, sir. Streptococcus agar-lactate. So it is group B streptococci. Am I audible, sir? Group
Tell,
B streptoc. Hello? Yes, sir?
tell me. Anything else you want to?
No. I'm just
No.
asking, am I audible?
Yeah, audible.
Oh, all right. So group B streptococci. It out of 10 to 30% of pregnant women are colonized with GBS in genital tract. It causes life-threatening infection in newborn infants as well as women. It can also cause serious diseases in pregnant women, elderly, and adults. With illness in newborns, GBS is the most common cause of sepsis and meningitis, and a common cause of this is pneumonia. GBS disease in newborn usually occurs in the first week. Its diagnosis and treatment. Diagnosis will include lab test of blood or spinal fluid. Treatment. Newborn and adults are usually treated with antibiotic which is given through intravenous system, that is IV. Enterococcus. So third organism is Enterococcus. Its habitat usually occurs found in intestine, female genital tract. 12 species, some of these are Enterococcus faecalis, Enterococcus faecium, durans, and avium. These are the common Enterococcus species. Normal inhabitants of Enterococcus is intestine. They cause UTIs in women. It will occurs in wounds, cholecystitis, blood infection, meningitis, bacteremia, subcutaneous and endocarditis, prostatitis. Enterococci are among the most common of nosocomial infections, especially in ICU patients. Streptococcus pneumonia. So its morphology, it is gram-positive diplococci with a capsule, which is a major virulence factor. Its habitat is nasopharynx of healthy carriers. It causes diseases like lobar pneumonia, meningitis, otitis media, sinusitis, bacteremia. These are the diseases which is caused by streptococcus pneumonia, and its transmission will happen through respiratory droplets. Laboratory features include alpha hemolysis on blood agar, bile soluble, opticin sensitive. Treatment will occur. Pathogenesis. So, Streptococcus pneumonia causes diseases. Will start with colonization. It will begins in the nasopharynx of healthy carriers. Invasions occurs when host defense are low, that is cold, influenza, immunosuppression patients. Capsule, the most important virulence factor, it inhibits phagocytosis. Pneumolysine, it damages epithelial cells and suppresses immune response. IgA protease, it helps evade mucosal immunity. Inflammation, it leads to consolidation of lung tissue in pneumonia. These factors explain why pneumococcal infections are more common in children, elderly, and immunocompromised individuals. So, its disease caused currently leading the cause of invasive bacterial disease in children and elderly individual. It causes 80% of lobar pneumonia, conjunctivitis, paranasal sinusitis, meningitis, all this acute and chronic bronchitis, septic arthritis, osteomyelitis, endocarditis, peritonitis, cellulitis, brain abscess. It is causes by due to streptococcus pneumonia. Lab diagnosis, they'll do direct microscopic examination of gram stain sputum. Smears will show the prevalent organism of streptococcus pneumococci. Sputum cultured on blood agar. Treatment will include pneumococci, which is sensitive to many antibiotics. Some isolates have recently been reported to resistant to penicillin. Vaccination, pneumococcal polysaccharide vaccine include for adults and elderly individual, conjugate vaccine for infants and children's. General measure, good respiratory hygiene, controlling chronic illness, avoid smoking. These measures reduce the burden of invasive pneumococcal disease. Prophylaxis and prevention, pneumococcal conjugate vaccine for infant immunization children, and this polyvalent polysaccharide vaccine for adults and elderly patients. Gram negative cocci. So they appear as red or a pink spherical bacteria after gram staining due to their thin peptidoglycan layer and outer membrane layer. The two most clinically significant genera are Neisseria gonorrhea and Neisseria meningitis of gram negative cocci. So important properties of Neisseria gonorrhea contain gram-negative cocci that resemble paired kidney beans. These two are the important gram-negative cocci, meningitis and gonorrhea. It is named after a German physician, Alexandre Neisseri. All Neisseria are oxidase positive, that is, they possess the enzyme cytochrome C. Neisseria meningitis, it causes meningococcal meningitis and septicemia. Both of these organisms are fastidious organisms requiring enriched media like chocolate agar and increased carbon dioxide for their growth. So they cause gonorrhea, STD in humans. Gonococci attacks mucous membrane of the gut, eye, rectum, throat, producing acute suppuration that may lead to tissue invasion, which is followed by inflammation and fibrosis. Gonorrhea is generally limited to superficial mucosal surfaces which lined with columnar epithelium. Its morphology, again, it is gram-negative kidney-shaped diplococci. Its habitat is human urogenital tract. Transmission is primarily through sexual contact, perinatal transmission to newborns, eyes, ophthalmia, neonatum. Significance, it causes one of the most common sexually transmitted infection, that is STIs. Its antigenic structure and virulence factor of Neisseria gonorrhea, pili or fimbriae, it aids in attachment to mucosal cell. Outer membrane proteins, OMPs, helps in adherence and immune invasions. Pro- proteins, protease degrades IgA on mucosal surfaces. IgA protease, it degrades IgA on mucosal surface. Antigenic variation, it enables immune escape and chronic infection. Diseases caused by Neisseria gonorrhea. So, urethritis in men, it is painful urination, purulent discharge. Cervicitis in women, vaginal discharge, pelvic pain, pelvic inflammatory diseases, ascending infection in females leading to infertility or ectopic pregnancy. Early recognition is key to preventing complications. So it is um, transmitted through sexual intercourse. It affects both male and females. Gonorrhea lasts 2 to 8 days after intercourse. Male gonorrhea is usually in the form of acute anterior urethritis. The organism invade the prostate, resulting in prostatitis, or extend to the testicle, resulting in orchitis. In females, it is in the form of cervicitis and urethritis with major symptoms which include mucopurulent, vaginal discharge, and mild lower abdominal pain. It may extend to the urine tube, causing salpingitis, fibrosis, and infertility. Asymptomatic males and females are major problem as unrecognized carriers of the disease. So about 50% of women with cervical infections are asymptomatic. It affects young girls through contaminated towels or toilet seats, causing vulvovaginitis. Cervical involvement extends through the uterus to the fallopian tubes, resulting in salpingitis, or ovaries, resulting in ovaritis. About 50% of women with uncomplicated cervical infection develop PID. Involvement of fallopian tubes or ovaries, resulting in sterility. It affects young girls through contaminated water towels, toilet. Oh, this is done. Ophthalmia neonatum. So, ophthalmia neonatum, it is an infection of the newborn acquired from birth through canal of gonorrheal mother. Mother will pass this disease to newborn infant. It involves cornea leading to, it can cause blindness. Occasionally, it is disseminated infection. Its diagnosis is acute urethritis in male, the urethral discharge examined by direct gram stain, presence of G-negative diplococci intra and extracellularly in pus cell. So in chronic male, acute and chronic female infection, in morning drop of prostatic discharge in chronic male infection, it causes Neisseria gonorrhea, cervical discharge, acute and chronic female infection. Here is the microscopic image of Neisseria gonorrhea gram stain of pure culture and Neisseria gonorrhea gram stain of a pustular exudate. So, direct gram stain usually hard to detect the organism due to the presence of normal flora or due to low net cultures done on chocolate or Thayer Martin media, which is incubated at 37 degrees Celsius in atmosphere of 5 to 10% of carbon dioxide present. Treatment and prevention. Treatment and prevention. First treatment, first line, it will include single dose intramuscular injection, often combined with azithromycin to cover possible chlamydial co-infection. It is resistant to penicillin, tetracycline, and fluoroquinolines. It is common. Prevention, safe sex intercourse practices, routine screening for high-risk individual, ophthalmia, neonatal prevention, erythromycin or tetracycline, eye drops for newborn infants. Neisseria meningitis. So its morphology is gram-negative kidney-shaped diplococci. Its habitat is again nasopharynge of healthy carriers. Transmission is through droplet infection and close contact. Epidemiology causes outbreaks in school, hostels, military camps. It is a major cause of bacterial meningitis in children as well as young adults. So it is highly contagious disease, usually occur in epidemics. It is in capsular polysaccharide shape, according to which meningococci are classified into groups. The most important groups associated with disease in men include A, B, C, Y, and W with 135 strains. Epidemics in Africa are mainly due to group A Neisseria meningitis. Properties of polysaccharide capsule: it enhances its virulence by its anti-phagocytic action. It is the antigen that defines the serologic group. It is the antigen which detect in the spinal fluid of patient with meningitis and in the vaccine that induces protective immunity. Meningitis. Meningitis, sudden onset fever, headache, neck stiffness, photophobia. Meningococci, that is septicemia, it is petechial rash, shock, multi-organ failure. Other forms include pneumonia, arthritis, urethritis. So these all will include in meningitis disease. It occurs in epidemics among young adults. Organisms occur in nasopharynges of healthy individual. Carrier rate is around 3 to 30% in healthy individual. During epidemics, the carrier rate reaches up to 80%. It starts in nasopharynx where it remains silent and give rise to exudative pharyngitis. From nasopharynx, the organism evade into the bloodstream, that is meningococcemia, causing high fever, hemorrhage, skin rash, etc. High fever. So the infection is transmitted through the droplets from carriers, healthy carriers, hemorrhage, necrosis of adrenal glands, that is Waterhouse-Friderichsen syndrome, characterized by high fever, shock, rash, widespread purpura, disseminated intravascular coagulation, and adrenal insufficiency. So this severe bacteremia, it is fatal due to circulatory collapse and suprarenal hemorrhage. So from bloodstream, the organism reaches meninges, causing meningitis. It is manifested by fever, severe headache, vomiting, nausea, rigidity of neck, stiffness, and back muscle pain. It may progress to coma within few hours if not treated properly. Diagnosis: CSF test. So specimens, that is cerebrospinal fluid, CSF, blood, nasopharyngeal swabs, all these specimens will include microscopy. They'll do gram-negative diplococci inside neutrophils in CSF. Culture media will use chocolate agar under carbon dioxide condition. Rapid tests include latest agglutination for capsular antigen. Molecular methods include PCR for rapid confirmation during outbreaks. Gram prophylaxis: rifampicin 600 mg. Overall, they'll give twice for 2 days. So vaccination, it will include polyvalent vaccine. For adults, it is effective in preventing epidemics of meningitis. The vaccine does not include group B polysaccharide, which is poorly immunogenic in humans. Trials are being made to prepare a recombinant vaccine. Gram-positive rods. So they appear as rod-shaped purple-colored bacteria after gram staining. They include both spore-forming, for example, Bacillus species, Bacillus cereus, Bacillus anthracis, Bacillus clostridium, and non-spore forming genera which include Corynebacterium, Listeria. It is generally found in soil, water, animals, and humans. Some of them are harmless, others are highly pathogenic. So this section will focus on medically important Bacillus species first. Bacillus, its characteristic: it is large in shape, gram-positive rod, aerobic or facultative anaerobic form. Endospores, it is highly resistant structures. Its habitat, it is widely distributed in nature, especially in soil. Pathogenic species include Bacillus anthracis, which causes anthrax, Bacillus cereus, which causes food poisoning and opportunistic infections. Non-pathogenic species, many use industrially, uh, for example, Bacillus subtilis, Bacillus anthracis. So its pathogenesis: Bacillus anthracis, its morphology, it is large rod with square ends arranged in chain or non-motile encapsulated, that is capsule made of polyglutamic acid. Habitat: its soil, infects grazing animals, that is zoonotic animals. Transmission through contact with infected animals. Products include inhalation, ingestion. So all this will include in Bacillus anthracis. Clinical forms of anthrax. Cutaneous anthrax, it is painless black estuary that is malignant pustule. Inhalational anthrax, it is wool sorter's disease, severe pneumonia with highly fatality. Gastrointestinal anthrax, so from ingestion of contaminated meat, it is rare but severe. Anthrax is nothing but primarily disease of animals which is caused in sheep, cattle, horses, causing septicemia and life-threatening disease which result in death. Humans accidentally contract this disease through infected animals or their products. Its lab diagnosis: samples taken from skin, sputum, stool samples, and blood samples, and smears. They'll prepare gram stain which demonstrate gram positive rectangular long bacilli which is arranged in long chain with clear zone representing the capsule. The polychrome methylene blue stain appears the organism blue in color while the capsule appears pink in color. Culture organisms grow on simple and enriched media. Animal inoculation in mice or guinea pigs, it is helpful to differentiate between anthrax and anthracoids. Serological test, it is participating and hemagglutinating uh, demonstrated in sera of men and animals. Treatment will include antimicrobial of choice. The most common one is penicillin, tetracycline, erythromycin. It is used with allergy to penicillin who have allergy. Prevention include disposal of animal carcasses by burning or deep burial in lime pits to avoid contamination, autoclaving of animal products, protective clothing and gloves for handling potentially infected materials, active immunization of domestic animals with life attenuated vaccines. Vaccine should be properly given to those animals. High risk of person immunized with a non-living vaccine. So proper vaccination in endemic areas, proper handling of animal products and human vaccine for at low risk workers. Public health relevance classified as potential agent. Bacillus cereus, it is motile, causes toxin-mediated food poisoning after consumption of cooked rice with enterotoxins. Toxin released by the bacteria lead to nausea, vomiting, and diarrhea, symptoms similar to SRS food poisoning. Food poisoning, two forms: emetic, that is vomiting, uh, and diarrheal, that is watery diarrhea. Opportunistic infection include endophthalmitis, wound infection, bacteremia, and transmission is through contaminated food. Spores survive cooking and germinate during storage. This endemic disease, it will cause symptoms like SRS, nausea and vomiting, diarrhea. Next is Clostridium. So its characteristics, again, it is gram-positive, spore-forming, obligate, anaerobic rods. Its habitat, it is usually found in soil, water, intestinal tract of humans and as well as animals. Pathogenic species include Clostridium tetani, Clostridium perfringens, Clostridium botulinum, Clostridium difficile. So these are the pathogenic species of Clostridium. Clostridium tetani causes tetanus, perfringens causes gas, gangrene, food poisoning, botulinum causes again food poisoning, difficile causes enterococci. So nearly 100 Clostridium species are identified, but only 25 to 30 species which are commonly cause human or animal infections. Its morphology: gram-positive long-chain bacilli with long terminal spores which give characteristic dumpstick appearance. They are motile in nature. Cultural characters: it is strictly anaerobes, grow on nutrient agar media on which colonies will be surrounded by clear zone of hemolysis due to its tetanolysine toxin. Organisms grow on Robertson cooked medium. Antigenic structure. So its antigenic structure produced by all toxigenic strain of Clostridium tetani, which are serologically identical. 10 types of antigenic variation among flagella are identified. Pathogeny, pathogenicity: humans, disease caused by tetanospasmin, which is neurotoxic endotoxin. Second is toxin are produced by vegetative cells of Clostridium tetani, and last is toxin production appears to be under a control of plasmid. Tetanus spasmin and tetanolysin. Tetanospasmin will act upon the CNS. It will release acetylcholine. Thus, it will interfere with muscular transmission. It blocks the release of inhibitory mediators, example glycine, which will lead to generalized muscular spasm. Tetanolysin, it is hemolytic toxin of minor importance in terms of pathogenesis of tetanus. Its infection will occur through contamination of wounds or with street dust containing spores, and local site of infection through spores germination. Injury: nail prick, surgical wound, gunshot wound, infected umbilical stump leading to tetanus neonatum. Germination of spores and development of vegetative organism. It will produce toxin which is aided by necrotic tissue, calcium salt associated with pyogenic infections. The disease is characterized by convulsive tonic contractions of voluntary muscle, spasm in jaw muscle which will lead to trismus lockjaw. Its diagnosis. So, tetanus diagnosis, many clinical, no laboratory tests will confirm it. Generally, wound culture are rarely positive. Differential diagnosis: trichin poisoning and hypocalcemia tetani. These will help in resulting the tetanus. So wound exudate are examined microscopically for the presence of gram-positive bacilli with drumstick appearance. The organism is generally identified by its pathogenicity to laboratory animals. Treatment: antitoxin and antibiotics will be given. Antitoxin given at once to suspected case without waiting for lab test diagnosis in order to neutralize the toxin before it fixes to CNS. Antibiotics, penicillin is the most common one, which is given in big doses to inhibit the growth of C. tetanus, that is Clostridium tetanus, and stops further toxin production. It also controls associated pyogenic infection. Prophylaxis: tetanus is totally preventable diseases. Active immunization: booster doses given to military personnel before or during war, pregnant women to guard against labor infection and to provide maternal immunity for the newborn. These injections will be given at the age interval of 2, 4, and 6 months. Passive immunization: antitoxin, it is given to wounded person without previous history of vaccination, and antitoxin prophylaxis should be accompanied by active immunization with tetanus toxoid. Clostridium perfringens. So, it occurs in deep wound infections with devitalized tissue, rapid onset, pain, swelling, cryptus. This will progress to necrosis and systemic toxicity. Diagnosis will be done through gram stain anaerobic culture. Treatment includes surgical debridement, high-dose penicillin, hyperbaric oxygen therapy. So it is the commonest membrane of genus Clostridium associated with gas gangrene, found usually in the colon around 25 to 35% of healthy individuals under certain condition, which will produce serious to life-threatening infection. Clostridium perfringens are of type A and C, which will produce enterotoxin, which are responsible for majorly food poisoning conditions. Morphology: it is gram-positive, large bacilli. They are non-motile species in genus. Spores are oval, subterminal, and non-projecting, and capsules are formed in tissues. Toxins or virulence factors. So, Clostridium perfringens produce variety of toxins and enzymes that will end up in spreading of infection. Alpha toxin, that is lecithinase, it acts on lecithin, which is a component of cell membrane. Theta toxin, it has hemolytic and necrotizing effect. DNase, hyaluronidase, and collagenase. Fourth is enterotoxin, which is produced by some strains of Clostridium. Type A and type C causes food poisoning following ingestion of warmed meat dishes. It will mainly cause when in the ingestion of uncooked or properly handled meat dishes, toxin is released in gut, which cause diarrhea after 6 to 18 hours of consumption, which will last for approximately 1 or 2 days. Gas gangrenes. Several species of Clostridia causes gas gangrene, but the commonest one is due to Clostridium perfringens. Pathogenesis. Infection occurs when wounds are contaminated with soil which contain the organism and its spores. Condition occurs in deep, lacerated, devitalized wounds, as of in car accidents or war wounds. The presence of foreign bodies, it will mixed with infection with aerobic pyrogen bacteria and decrease blood supply. It will lower the oxygen level, which will favors in terms of germination of spores. Gas gangrene, it is the multiplication of vegetative cells which will ferment sugars by producing gas. The tissues will interfere with blood supply, which will lead to tissue death. Necrotizing toxin, collagenase and hyaluronidase will favor necrosis and helps in the spreading of infection. Proteolytic Clostridia digest dead tissues, which will again lead to change in color and foul odor of wound. This all will be accompanied by generalized toxemia, which is fatal. Diagnosis is primarily on clinical grounds, bacteriological confirmation from wound exudate and swabs from deeper areas. Direct smears are stained through large gram-positive rods. Prevention depends upon educate cleaning, hygiene, contamination of wounds, surgical removal of foreign bodies, and excision of all devitalized tissues. Administration of antibodies, especially penicillin, common one, antitoxic sera for passive prophylaxis, and toxoids which are not available for active immunization. Clostridium botulinism. So, Clostridium botulinism, it is gram-positive in nature, spore-forming aerobic rod. Its habitat is soil, sediments, improperly processed canned food. Significance: produces the most potent neurotoxin known as botulinum toxin. Mode of interaction, you can tell ingestion of preformed toxin, that is foodborne colonization in infants, intestines, infant botulinism, or wound contaminations. Pathogenesis: toxin blocks acetylcholine release at neuromuscular junction, so it will lead to paralysis. Foodborne botulinism, nausea, vomiting, diplopia, descending paralysis. Infant botulism, floppy baby syndrome. And last is wound botulinism, which is similar to foodborne but forms spore germination in wounds. Cultural characteristics: wound botulinism and intestinal botulinism. Wound botulinism can occur in rare cases where devitalized tissue is contaminated with soil. Intestinal botulinism in infant, which is known as floppy baby syndrome, is due to ingestion of spores followed by germination and production of toxin in gut. So the feeding of honey has been implicated as possible cause of infant botulinism. Botulism toxin uses: medically, treatment for dystonias, chronic migraine, hyperdiaresis; cosmetic, Botox to reduce wrinkles by temporary muscle paralysis. Diagnosis: toxin can be demonstrated in serum. Antigenic type of toxin identified by specific antitoxin is in mice. After this, mice will die rapidly. Botulinum growth from leftover food and tested for toxin production. It is rarely done. In infant, Clostridium botulinum and toxin can be demonstrated in stool but not in serum. Toxin demonstrated by passive hemagglutination or RIA, that is radioimmunoassay. Treatment is through rapid administration of polyvalent antitoxin, and prevention is careful sterilization of food before canning. Clostridium difficile. Clostridium difficile is again gram-positive spore-forming anaerobic rod. Its habitat is normal intestinal flora in some individuals. Disease, it will call antibiotic associated diarrhea and pseudomembranaceous colitis, that is PMC. Risk factor, prolonged broad spectrum antibiotic use. So its pathogenesis is associated with toxins, toxin A, enterotoxin, and toxin B, cytotoxin. Important properties, it is non-spore forming, non-acid fast gram-positive bacilli which will clubbed with wider at one end and are arranged in parisates or in V or L-shaped formation. Their arrangement resemble Chinese letter. They are aerobic or facultative anaerobic, non-motile in nature, and catalase positive organisms. Although many species of Corynebacterium can function as opportunistic pathogen, but the most commonly associated human pathogen is Corynebacterium diphtheria. Its pathogenesis, it produces toxin. Lysogenic beta phages are able to produce diphtheria toxin because phage carriers the toxin gene. Major significant virulence factor produced by diphtheria is diphtheria toxin, which is entirely under the genetic control. As I mentioned earlier, toxin A, enterotoxin, and toxin B is cytotoxin. Enterotoxin, fluid secretion, diarrhea will occur, and cytotoxin, mucosal damage and formation will happen. Then epidemiology. Humans are the only natural host for Clostridium diphtheria. Aerosol droplets dissemination spread the organism from person to person. It is rarely by direct transmission. Organism can also infect skin, especially through tropics, which can occur worldwide in person with poor skin hygiene. So maintaining hygiene is the important property to avoid infection with Corynebacterium diphtheria. Disease, most prominent sign of diphtheria is thick gray adherent pseudomembrane over the tonsils and throat. There are three prominent complications: extension of membrane into larynx and trachea, causing airway obstruction; myocarditis accompanied by arrhythmias and circulatory collapse. Laboratory diagnosis through microscopy, culture, and serological test. So, throat swab culture on Loeffler's serum slope, Albert stain, metachromatic granules, and toxin detection, that is ELKS test. These three tests will be done for laboratory diagnosis. Treatment is through diphtheria antitoxin and antibiotics, which will include erythromycin or penicillin. Prevention, DPT, vaccination. Important properties include the genus Listeria. It is a small gram-positive, non-sporing, forming facultative anaerobic bacilli. Its characteristic is gram-positive, motile, facultative intracellular load. Its habitat is soil, water, decaying vegetation, dairy products. Significant, it is an opportunistic pathogen affecting neonates, pregnant women, elderly, and immunocompromised individuals. So, species of medical importance, that is Listeria monocytogenes. It causes neonatal sepsis and meningitis, maternal listeriosis, flu-like illness, which also cause abortion, meningoencephalitis in elderly and immunocompromised individuals. So it invades intestinal epithelial cells and it went into bloodstream, which causes blood-brain barrier or placenta. Listeriorizine, it helps escape from phagocytosis and it aids in intracellular movement. So infection during pregnancy can cause abortion, premature delivery, or sepsis during period. Meningitis or sepsis can occur in immunocompromised or immunocompetent individuals. Gastroenteritis, it is caused by the consumption of contaminated as well as dairy products. Undercooked meats is a major health problems. Laboratory diagnosis, culture, blood CSF on blood agar will show the presence of Listeria bacteria. Its motility test is umbrella-shaped growth and room temperature. Serology, that is PCR, in severe cases will show the result. Treatment is again with ampicillin or with gentamicin is the most common antibiotic given in cases of listeria. Prevention and control, limited exposure of immunocompromised patient to potential sources such as infected animal and their products and contaminated vegetable. There is no vaccine available up to date. Gram-negative rods. So gram-negative rods will occur pink or red rods after gram staining due to thin peptidoglycan and outer membrane containing lipopolysaccharide. Its families will include Enterobacteria, Pseudomonas, and Vibrio, etc. So its clinical importance is it will cause a wide range of infections including UTI infection, gastroenteritis, and pneumonia, as well as sepsis. So the intestinal tract of humans and animals is the common habitat. It is in gram-negative facultative anaerobic bacilli. About 32 genera is identified, which contain more than 130 species. Some genera which is well known, example include Escherichia, that is E. coli, Shigella, Salmonella, Enterobacter, Klebsiella, Serratia, and Proteus, Mirabilis, etc. Some enteric pathogens, example E. coli, are part of the normal flora and accidentally it will cause diseases. Others include Salmonella and Shigella, which are regularly pathogenic for human. Its morphology, it is gram-negative bacilli, non-motile and or motile by trichous flagella. It is non-spore forming and some are encapsulated. E. coli, that is Escherichia coli, normal inhabitants of intestine of men as well as animals. It will cause disease in human. Its morphology, gram-negative bacilli, motile, some strains are capsulated. Cultural characteristic, it is facultative anaerobes. It will grow on simple media. On McConkey media, they'll produce rose pink color colonies due to lactose fermentation. Its virulence factor, many strains of E. coli have powerful toxin which is responsible for causing diseases. Pili, it is an adhesive factor. Disease caused by E. coli, most common diseases UTI, urinary tract infection, which will majorly occurs in women, intestinal disease. So UTI, it is the common cause. Uropathogenic E. coli colonize the vagina and periurethral region from where they ascend to the bladder or kidney, which causes cystis or pyelonephritis. Intestinal disease, different strains of E. coli can cause diarrhea through different mechanisms. Enterotoxigenic. Enterotoxigenic, it contains serotypes which will cause infantile diarrhea. Diarrhea is watery and ranges from mild to severe cholera and it may be fatal. Organism adheres to intestinal epithelium through pili or colonization factor which will liberate enterotoxins. The toxin, this will cause diarrhea by stimulating in intestinal mucosal cells. Second is enteropathogenic E. coli. So certain serotypes, example O55, O111, this will cause outbreaks of neonatal diarrhea in nurseries. They act mainly by adhering tightly intestinal mucosa, which will result in loss of microvilli and cupping of cells around the bacteria. So this thus preventing the normal functions of absorption and secretion. Third is enteroinvasive E. coli and fourth is enterohemorrhage. So invasive, again certain serotypes will include which will cause dysentery like diarrhea through invasions of intestinal epithelial cell. Enterohemorrhage, these strains called hemorrhage which is known as colitis which will form certain types of diarrhea with bloody discharge will produce Shigella toxin. Gram-negative ROS, neonatal meningitis. E. coli causes more than 40% of neonatal meningitis, followed by group B streptococci. It causes bacteremia and endotoxic shock in immunocompromised, immunocompetent individuals. It causes hospital-acquired infection. Diagnosis, Samples include urine, pus or stool samples, CSF, etc. Pathological material is inoculated on media, McConkey agar. LF colonies are further identified according to their morphology and biochemical infection reactions. Enterobacter. Enterobacter species, it is occurs as indigenous intestinal flora. Habitat is soil, water, intestinal tract. It is an opportunistic infection which causes UTIs, respiratory infections, septicemia. It is motile, lactose fermenting, part of anterior bacteria. Its treatment offered multi-drug resistant, used for serious infections. It is similar to E. coli except in being citrate positive. It is antigenically resembling the Salmonella species. It is seen as opportunistic pathogen in immunocompetent individuals. It will cause UTI, sepsis, wound infection, osteomyelitis, and gastrointestinalitis in elderly hospitalized patients. Cases of neonatal meningitis has been reported. Species include for Citrobacter is Friondi and Kosari. So in today's class, we studied about gram-positive cocci, that is Staphylococcus, Streptococcus, and Enterococcus. Gram-negative cocci, that is Neisseria gonorrhea and Neisseria meningitis. Gram-positive rods, that is Bacillus, Clostridium, Corynebacterium, and Listeria. Gram-negative rods, Enterobacteria, and other species. Its morphology, virulence factors, disease causing, diagnosis, is treatment as well as prevention. Gram positive and negative cocci, gram positive and negative rods. These two we studied in detail today. So for question and answer part, for 3 marks question, define coagulase and mention its significance in Staphylococcus aureus identification. Name 2 enzymes produced by Clostridium perfringens. Third, what is the causative agent of diphtheria and its primary virulence factor? Fourth, mention 2 clinical features of botulinism. Fifth, what is the role of M protein in Streptococcus pyogenes? Sixth, define Staphylococcus aureus. Seventh, list the 2 types of hemolysis, alpha, beta, gamma. Any two you can mention, majorly alpha and beta. Eighth, name the causative agent of tetanus. Ninth, mention 1 toxin produced by Clostridium diphtheria. 10th, write 2 diseases caused by Listeria monocytogenes. For 5 more questions, explain the laboratory diagnosis of S. aureus, discuss the pathogenesis and prevention of Corynebacterium diphtheria. Third, describe clinical forms of anthrax. Fourth, write a short note on Clostridium difficile and its management. Fifth, explain the virulence factors of Neisseria meningitis. Sixth, explain the pathogenesis of S. pyogenes. 7. Discuss virulence factor and laboratory diagnosis of Neisseria gonorrhea. 8. Describe clinical features and prevention of tetanus. 9. Write a note on anthrax with its type and management. And last is discuss laboratory diagnosis of lobar pneumonia caused by S. pneumonia. So that's all for today's class.
Yeah, very nice presentation, madam.
Thank you, sir.
So you have covered complete bacteriology, I think so.
Sir, few bacteria is remaining, that is
Yeah,
Klebsiella, Shigella, Salmonella, Vibrio. These are
yeah,
pending. I just covered gram positive and negative cocci and rods in today's class.
yeah,
In next class, inshallah, I'll cover the whole
yeah. Thank you.
system.
Thank you so much, madam.
Thank you,
Thank
sir.
you. Thank
Thank
you.
you.
Thank you.